Thank you for the introduction. Hello everyone, and welcome to my talk. I'm very excited to be here today in Madison, presenting on a tool we've developed at the Pacific Northwest National Laboratory to simplify the process of sequence-based protein function characterization. And here I will present Snekmer, which is a scalable pipeline for protein sequence fingerprinting using an amino acid recoding approach. Now, of course, there already exist currently methods for protein function annotation with which many of you are already familiar. Commonly, a sequence similarity search a la BLAST, or the basic local alignment search tool, is performed across a genome set. Alternatively, sequences can be annotated via multiple sequence alignment followed by comparison against hidden Markov models, or HMMs for short, using tools such as Hammer. However, these tools involve pairwise or multiple sequence alignment, which can be resource intensive, but most importantly to us, the scientists, time consuming. Additionally, appropriate profile HMMs can be difficult to source and build, again, adding up to more time and more effort. One solution has been the use of k-mers, which simplify how sequences are represented. In k-mer-based approaches, sequences are represented by counting the individual subsequences of length k, which comprise the total sequence. Advantages of this approach are the ability to capture similarities between partial or distantly related sequences. Additionally, the k-mer representation is simple, and thus computation is relatively speedy. The end result, a vectorized representation of a sequence, is also in the form of a vector, making it transferable to computational efforts that incorporate exciting new developments in areas of machine learning. However, while k-mers are frequently used in genomics to represent DNA or RNA sequences, a key disadvantage in invoking the k-mer approach in protein sequence analysis is that there are 20 amino acids. Thus, k-mer-based representations can quickly scale to out-of-hand proportions, particularly for longer values of k. To address this issue, we have developed alphabets to recode amino acids, a process we have termed amino acid recoding, or AAR. Through amino acid recoding, amino acids are grouped by various similarity measures, such as chemical properties. We have also incorporated recoding via substitution matrices, such as MIX, which stands for Matrix to Improve Quality and Similarity Search. The alphabets we explored are further illustrated on the slide, and some references for literature sources are also mentioned at the bottom. In building our AAR k-mer-based protein function annotation tool, we envisioned a modular workflow combining these approaches in a manner well-suited for high-throughput data analysis. So at a high level, our workflow follows these steps. First, sequences are recoded according to the desired AAR scheme, or alphabet. Next, substituent k-mers are counted for each individual sequence, and the resulting k-mer frequency vectors are built. Finally, these vectors are used to model sequence similarity using various methods. Our tool, Snekmer, implements these steps as a modular pipeline. Snekmer is a fully open source, publicly available software package, which is written in Python, and thus benefits from Python's rich ecosystem of bioinformatics and data science libraries, including those shown here on the slide. Snekmer, as some of you may have guessed, also derives its name from the Python-based workflow management system called Snekemake, which underlies the steps of the pipeline. Snekemake offers several advantages, including innately imparting reproducibility via self-contained workflow steps, enabling scalability across environments from single to multi-core cluster or cloud computing environments, and allowing for modular deployment of the individual processing steps. Snekmer executes our workflow via self-contained modular steps, known as rules for those familiar with Snekemake. First, Snekmer detects and handles input sequence files before passing them on to the next step, which executes the amino acid recoding and so-called k-merization of the sequences. The user can then select one of two distinct flavors of Snekmer. Snekmer model, shown on the top, trains supervised machine learning models based on existing annotations via determination of k-mer-based probability scores. The bottom workflow shows Snekmer cluster, which implements one of several available clustering algorithms to build unsupervised clusters based on the AAR K-mer vector profiles. A third available mode, Snekmer Search, is able to take the pre-trained models from the Snekmer model step and use these to annotate unlabeled sequences. The end result is family probability scores for whether a given unlabeled sequence belongs to a protein family based on its AAR K-mer profile. To validate our pipeline, we evaluated Snekmer's performance using a curated set of pre-annotated protein sequences that are involved in the nitrogen cycle. The cycle and involved families are illustrated in the figure on the right. The data consists of roughly 33 families with 5,500 total sequences pre-annotated using hidden Markov models. We also manually differentiated between subfamilies for 3 families that were divergent in terms of activity. We then evaluated the performance of both the Snekmer model and cluster modes in reproducing these families. We ran Snekmer model across all 6 AAR schemes, plus once with no recoding, with varying values of K. The models were evaluated using 5-fold cross-validation. The mean performance of all the resulting protein family classification models is summarized here via the area under the receiver operating characteristic curve, or AUC-ROC for short. For those of you who are unfamiliar with the AUC-ROC metric, it is a measure summarizing the true positive rate versus the false positive rate of a binary classification model. An AUC-ROC score of 1.0 corresponds to perfect classification, whereas a score of 0.5 is equivalent to random chance. We found that the average AUC-ROC scores exceed 0.9 in more than half of alphabet and K combinations, thus closely mirroring the performance of the HMMs. We also found that the AAR K-mer approach can match or even outperform the K-mer-only approach at classification, particularly for longer K-mer lengths, and that a diverse range of alphabet and K configurations produces good classification models, here defined as having an AUC-ROC score of greater than 0.9. Furthermore, we discovered an interesting phenomenon with the different alphabet and K configurations. If you examine the figure on the right, you'll notice a sort of Goldilocks zone at moderate alphabet complexities and values of K where the models perform best. This makes sense, as you can imagine that when an alphabet is minimally complex and K-mers are short, the amount of information captured by these K-mers is low. Indeed, these classifiers perform no better than a random coin flip. However, as we increase the alphabet complexity and K-mer length, we access the optimal combination of a sufficiently complex alphabet with K-mers that are long enough to capture family-to-family sequence variations, as well as the unique K-mer signatures of a given family. Finally, when we move past this regime towards complex alphabets with longer K-mers, the models are now trained on very long and varied K-mers. These long, complex K-mers are not representative of the total sequences in the family. Thus, we begin to see a decrease in the performance of the classification models at higher K and alphabet complexities. Similarly, we ran Snekmer cluster mode on the nitrogen cycle families and found that Snekmer-generated clusters also work to recapitulate the known families. The clusters are visualized on the left, and the corresponding AUC-ROC heat map is shown on the right and demonstrates a similar Goldilocks trend as I described with the Snekmer model mode. So in summary, we've developed an AAR K-mer tool called Snekmer and demonstrated that its operating modes can recapitulate known protein families using an example data set of nitrogen cycle families. We've now submitted this work for publication. It's currently under review, and our fully working prototype, including example usage and extensive documentation, is available on our GitHub repository shown here. We have many future plans for Snekmer, which I've delineated here, including further code optimization to facilitate its usage across larger sequence sets and integration into user-friendly web applications, as well as K-base. So with that, I'd like to thank all of my wonderful colleagues without whom Snekmer would not have been possible. In particular, Jason McDermott, who came up with the initial Snekmer and AAR K-mer combined approach and who really led this work, and Bill Nelson, who drove the application-based side of Snekmer. And lastly, I'd like to thank all of you for your time and attention. So with that, I don't think we have time for questions, but I'm happy to address them via email. Feel free to contact me or stop by my poster tomorrow afternoon at the exhibit hall. Thank you.